The next speaker is going to be Dr. Helmut Hillebrand, who recently moved from Cologne in Germany to Oldenburg. Like Dr. Moran, he's been working on this subject of biodiversity and ecosystem function for quite some time. I myself have quoted a lot of his work lately. And without further ado, I'll ask him to come up and speak, where his title is Biodiversity Effects in Natural Ecosystems, Maturation of a Paradigm Shift.
Thank you, Jed, for introducing me. Thank you for coming. So I will kind of change a little bit the perspective organism-wise and talk about a little bit larger stuff which have many cells sometimes. And if you look at a textbook sketch of how diversity is regulated in ecosystems on regional and local scales, what you see is that human actions interfere with almost all of these processes. We change dispersal because we fragment landscapes, we transport species around the globe. We change environmental conditions because we change temperature, CO2, pH. We change predation because we add or remove consumers. And we change competition because we change nutrient supply and alter nutrient ratios in a global biogeochemical perspective. Obviously, this changes diversity, and obviously this leads to a reduction in diversity in many ecosystems. So the obvious question is, what does this mean for ecosystem function? And with function in this talk, I will mainly look at ecosystem process rates. But we could also look at states like stability, or we could look at ecosystem use, goods and services we derive from ecosystems. This is a true paradigm shift because diversity has been dealt with as a response variable in the majority of ecological literature until like 15 years ago. So we have seen diversity as responding to properties and processes of ecosystems. Now we realize that there is a feedback that diversity might actually affect ecosystem properties and processes. And the kind of experiments Peter has talked about have been very informative to disentangle this feedback. So here is a summary of these experiments published 3 years ago in Nature by Brad Cardinali and co-authors. And they found that compared to the mean single species, diverse mixtures have a higher standing stock and they deplete their resources more consistently than these monocultures. And this is independent of whether you look at plants, herbivores, predators, or detritivores. And they also show in that paper it's independent whether you look at terrestrial, freshwater, or marine systems. In the same paper, they show that if you look at the best performing single species, the pattern doesn't remain as clear. Compared to the best monoculture, the diversity mixtures sometimes do better, but not in a completely consistent way. So this seems to be kind of an agreement on this debate. And you could think we could kind of close this case, but you then see papers like this by James Grace et al. In Ecology Letters 2007. They made structural equation models looking at the effect of abiotic environment and disturbance on richness and biomass, and also the effect of richness on biomass production and productivity on richness. And in all the systems they were investigating, you see that they found no significant effect of richness on any of these, in any of these grassland systems. So they kind of conclude that in natural systems, the abiotic environment and disturbance regimes are much more important for ecosystem processes than the diversity of the assemblage. And actually, if you look at it from a theoretical viewpoint, you see that, of course, diversity is both a response and a driving variable. We know that if we increase resource supply, we change richness and biomass. Species energy theory is one example for that. Resource ratio theory predicts that if we change the stoichiometry of resources, we change richness and biomass. And then, as a new addition, we have evidence that changing richness changes biomass. So, in a real system, productivity as an ecosystem process and diversity are entangled in a web of interactions and driven by different processes. This leads to the question whether the simple experiments which we did on biodiversity ecosystem functioning actually tell us something about the real world. Even a simple real world like this rock pool, do we catch their dynamics and their processes with our experimental systems? And how do we deal with the complexity of natural systems in this issue? So as soon as you start thinking about complexity, the way to go is to philosophy. And here is advice from some famous philosophers. The world is a complicated place, Hobbes. Whenever it seems that way, I take a nap in a tree and wait for dinner. The second way of dealing with complexity is adding complexity to our experiments. And that is actually what I want to talk about in the rest of my talk. We have this feedback loop between ecosystem states and properties on diversity and on from diversity to processes in ecosystems. And does this picture change if we make systems more complicated, if we add this natural complexity to our experiments? And I will start with looking at adding spatial dynamics. And the reason for that is that spatial dynamics are very important for understanding the coexistence of species in the first place. Many systems are connected by dispersal, and the coexistence of species in any of these high latitude lakes here is dependent on the input of organisms from the other parts of this meta-community. So meta-community dynamics allow regional coexistence, and there are a number of different mechanisms how this coexistence actually takes place. If you look at theoretical approaches to this topic, you see here a paper by Leroux et al., where they, in the model, changed dispersal rates in a meta-community. And what they found is that they have this hump-shaped curve of local diversity with highest local coexistence at intermediate dispersal rate. And in their model, this was also, at these intermediate dispersal rates, they also found the highest productivity. In an experimental system, based on different mechanistic approaches, we still find the same pattern. Increasing dispersal leads to a hump shape of final local species richness of a benthic microalgal community, and this is also where we find highest biovolume production. This is again a tiny microbial system, and you already have seen this paper by Plachnik et al. You can also see this pattern in nature. And here we have a graph of resource use efficiency, which is the build-up of biomass per unit resource along a gradient of genus richness of phytoplankton in a large Scandinavian data set. And you see that there is a strong positive interaction, strong positive relationship between the two. The second thing I would like to talk about is trophic structure, and that is a point where aquatic ecology has actually played a major role because most of the terrestrial experiments, as Peter already said, have been on one trophic level. However, we know that from the record of extinctions, that most of the extinctions are on the consumer level or on the predator level, whereas most of the invasions are not from these groups, which means that the net diversity change for predators and consumers is much more negative than for autotrophs. So what does this mean in an ecosystem if we lose consumer species? This is one of the a series of experiments done by the group of Emmett Duffy. They manipulated richness of grazers in a seagrass system, and they found that with decreasing grazer species richness, you find decreasing grazer biomass, increasing algae biomass, no change on eelgrass biomass, and losing diversity in the grazer system increased benthic diversity. So there were obviously a number of processes allocated to the species richness. In a recent study by O'Connor et al., you see that it was not diversity which changed the pattern, but actually a certain species. So here we have a simple food web with 3 predators, 3 crabs, and their main prey, oysters. And O'Connor et al. Reduced the richness of this assemblage by deleting certain species and looked at oyster survival as a response variable. And you see that the significantly different treatments were all those where the blue crab was deleted. So it was not the richness which was responsible for a certain function, but it was the identity of a certain species, the identity of the blue crab. If that species was present, the predation pressure was high. You could also look at what is the effect of prey diversity on the consumer, and you can do this in controlled experiments, but you can also do it by looking at a set of experiments which differs in their species richness. And Brett Cardinale and I did this meta-analysis, which has the advantage that we go up to 70 species of algae. And actually there was significant effect of algal species richness on grazing. The fewer algae we had in the system, the stronger was the grazing pressure. The grazer effect size increased with decreasing algal species richness. And of course, this is a very cloudy picture and a very scattered diagram. But if you control for a number of variables, you can see that both the number of species and the evenness in this algal assemblage had significant effects. And actually, if you eliminated either or from the system, the models became much less parsimonious than the full model. So actually, when we control for other variables, we still find a strong effect of prey richness on consumer pressure. This leads to the third point I would like to address, and that is the point of evenness and dominance. Because we deal with this topic because biodiversity is lost in ecosystems. But actually, before we lose a species in an ecosystem, we change dominance structure. Getting a species to complete extinction is a very, very long process, but changing the dominance structure is a very fast process. So actually, we change the evenness and dominance in these systems and thereby change the distribution of traits in an ecosystem. And that may affect ecosystem processes. It actually affects the survival of species and coexistence. And this is one example where people have dealt with this aspect of dominance. This is a very nice paper by McIntyre et al. They looked at recycling of nitrogen and phosphorus in Lake Tanganyika by fishes. And from species-specific data they had on these fishes, they could simulate the extinction of species and calculate the consequence for recycling rates. And they did this in a random model, so they randomly deleted species, and you show that there is, with decreasing richness, there's a strong decrease in N and P recycling. If you delete species by rarity, you see that we have kind of a redundancy at the beginning where not much happens, but then we get to a very rapid decrease both in N and P recycling too. This effect of dominance has also an important consequence for spatial dynamics, because you can easily see that if you have a meta-community consisting of 3 habitats, and the 3 habitats are dominated by different species, we have a very efficient spatial rescue of these species in the regional species pool, because all of them contribute to the propagule pool, and all of them may be retained in the meta-community. However, if you have a meta-community where the dynamics are so synchronized that the same species becomes dominant in all patches of this meta-community, you actually will have only this species as the main part of the propagule pool, and this rescue effect may actually collapse. This leads to the acknowledgement that we completely understudied the problem of beta diversity, the diversity between habitats. What is the importance of species turnover? And my first hunch would be that with increasing beta diversity, we might actually reduce the mean process rate on the regional level, but increase the stability of a process. There's a session adjunct to this plenary, and this afternoon Robert Ptachnik will present new data talking about beta diversity effects and the role of regional dynamics for biodiversity ecosystem function relationships. So if you are interested, please go to that talk. Finally, almost all our experiments, and some of the papers Peter presented are actually an exception, but most experiments deal with one aspect of diversity and one function: changing richness, looking at productivity. However, a real ecosystem, of course, is characterized by a multitude of functions. And we did a kind of back-on-the-envelope calculation to address this problem. So if you increase the number of functions you're looking at, and if you consider that different species are carrying out major aspects of these different functions because they differ in their traits, then you need more species to sustain more functions compared to the classical saturation function which we see here. And we used the Seagrass experiment by Emmett Duffy to make this point. If you look at one function at a time and you decrease species richness in this system, you have a decreasing probability of sustaining this function, but even at only one species you still have a decent probability of having this function sustained in this by this assemblage. However, if you increase the number of functions you're looking at, this probability gets lower and lower. And our feeling is that the mainstream type of biodiversity ecosystem functioning experiment has actually underestimated the consequences of